Hi guys, in the news you will be listening that coronavirus is a genetically engineered virus and it escaped from Wuhan Lab. But the analysis of public genome sequence data from SARS coronavirus 2 and related viruses found no evidence that virus was made in laboratory or otherwise engineered. Christian Anderson said, by comparing the average genome sequence data for known coronavirus strains, we can firmly determine the SARS coronavirus originated through natural processes. She is an associate professor of immunology and microbiology at Scripps Research. Coronavirus are the large family of viruses that can cause illnesses ranging widely in severity. The first known severe illness caused by coronavirus emerged with the 2003 severe acute respiratory syndrome epidemic in China. A second outbreak of severe illness began in 2012 in Saudi Arabia with the Middle East respiratory syndrome. The scientists analyzed the genetic template for spike proteins armatures on the outside of the virus that it uses to grab and penetrate outer walls of human and animal cells. More specifically, they focus on two important features of the spike protein: the receptor binding domain, a kind of grappling hook that grips onto host cells, and the cleavage site, a molecular can opener that allows the virus to crack open and enter host cells. The scientists found that the RBD portion of the SARS coronavirus 2 had evolved to effectively target a molecular feature on the outside of human cell called ACE2. That is a receptor involved in regulating blood pressure. The SARS coronavirus 2 spike protein was so effective at binding to human cell, in fact, that the scientists concluded it was a result of natural selection and not the product of genetic engineering. This evidence for natural evolution was supported by data on SARS coronavirus backbone as well. Its molecular structure shows that if someone were seeking to engineer a new coronavirus, a pathogen, they would have constructed it from the backbone of a virus known to cause illness. But the scientists found that coronavirus 2 backbone differed substantially from those of already known coronavirus and mostly resembled related viruses found in bats and pangolins. Anderson also said these two features of the virus, the mutation in the RBD portion of the spike protein and its distinct backbones, rules out laboratory manipulation as the origin of SARS coronavirus 2. Josie Golding, PhD epidemics lead at UK-based Wellcome Trust, said the findings by Anderson and his colleagues are crucially important to bring an evidence-based view to the rumors that have been circulated about the origin of the virus. They conclude that the virus is a product of natural evolution, ending any speculation about deliberate genetic engineering.